Welcome back, everyone, to the BiochemVerse by Hammad to the drug discovery and designing series. And sorry for little bit vacations, and that because I was a little bit sick and ill. Distinguish these words as you are on. So today's lecture is the molecular docking basics, tools, and computational workflow. Today, we are going to walk through what molecular docking is, why it is used, and how to practically perform docking using commonly available tools and web server. Molecular docking is one of the most fundamental techniques in computational drug discovery. It helps us predict how a small molecule, you can call it as a ligand or a lead molecule, interacts with a biological target such as protein or enzyme, as we have discussed before in our lecture, target identification and validation. If you don't listen to haven't listened to that lecture, then go first listen to it and then come here. So we move our lecture. As I have started initiatory lecture as coming introducing and then started lecture, no extra talks, because we are too late in our previous lectures because we have missed so much days. So it's basically a ligand molecule interacting with a biological target, which may be protein or enzyme or any nucleic acid also. Think of docking as a virtual experiment where we simulate the binding between molecules before testing them in the lab. This saves tremendous time and cost and narrows down the best candidates. The best candidate we are doing, if there is post announced, they conduct a written test before recruiting anyone. On recruiting on getting the written test, they narrows down the best candidates, then interview them and pick the best one. So by the end of this lecture, you will confidently understand docking concepts, ligand and protein preparation, how scoring functions work, which tools and servers are used, step by step use of these tools. I'll make sure that use of the tools, the workflow of molecular docking, I'll also add up a video in it in this lecture, or separately I upload the video of uh, step by step guide to them. So moving forward, the what is molecular docking, we have discussed about this, and I have to again was also discussing, we have to discuss the molecular docking still. Molecular docking attempts to predict the best way a ligand fits into a protein's active site. In simpler terms, it estimates how strong the binding is, where the ligand binds, how the ligand site inside the pocket sits inside the pocket, the binding pose, and the binding affinity and binding site. These three are terms we can refer. So take it in easy way, that how strong the binding is, we can get it by the binding affinity. Where the ligand binds means the binding site, where the ligand has made bond with the protein or the biological target, and how the ligand sits inside the pocket means the binding pose, that from which terminal or which site it has made bond with the target. So imagine trying to park your car in a parking spot. The parking spot is the protein active site and the car is the ligand. If the parking space is too tight, poor fit. If the car aligns perfectly, strong binding. If the angle is wrong, the car may be inside but not ideally positioned. Docking is the computer's way of checking which car fits best and which position is optimal. So tool guidance, common tools that can be used for docking include AutoDock and AutoDock Vina, PyRx for beginners, and SwissDock and DockThor also the web-based docking servers. In the practical workflow, we typically start by driving protein structure from the protein data bank and ligand from the PubChem or Zinc libraries, as we have discussed in our introduction to drug discovery and designing. So, the principles behind molecular docking. Docking is based on the idea that the protein and ligand must complement each other. The best orientation has the lowest energy, and the last one is the scoring function approximates real life biological bindings. So, imagine inserting different keys into a lock. Some keys don't enter at all, that would be the poor match, and some keys enter but don't turn, that's a partial match. One key fits and turns smoothly, that's the perfect match. Docking algorithms search through many poses of the key until they find the one that best fits. So tools and algorithms, docking tools uses search algorithms and scoring functions. Some of the search algorithms, you take examples like Lemakin's genetic algorithm, that's also, and the scoring function like binding free energy calculations. So when using tools like AutoDock Vina, the scoring functions gives a value like minus 6.0 kilocalories per mole or minus 7.5 kilocalories per mole. Lower is stronger binding. The lower one is minus 7.0 kilocalories. That was the example. So the core principle, lock and key or induced fit concept, search algorithm, scoring function, and protein ligand complementarity. So moving next, types of docking. Docking can be performed in different modes: rigid docking, semi-flexible docking, flexible docking, blind docking. So rigid docking, both proteins and ligands are stuff. Useful when the structure is known and doesn't change. And a semi-flexible docking is like protein rigid, ligand flexible, most commonly used. And flexible docking, both protein and ligand flexible, more accurate but computationally expensive. So blind docking, when you don't know the binding site, you cover up whole protein, your whole target, and give it to the dimensions to the software that you find the binding poses in this dimension. So you blindly give the whole structure of protein and ask them to find the binding poses in that specific pose, not only that specific site, not only you have to find it blindly in the whole protein structure because we don't know the. So the entire protein is scanned in the blind docking. Think of trying to wear shoes. Rigid docking like wearing stiff boots, no flexibility. And semi-flexible, wearing sneakers. I like them sneakers firstly, but nowadays I'm liking the formal shoes like Derby, Chelsea. So semi-flexible, wearing the sneakers, the shoe adjusts a bit, and flexible docking, wearing soft socks, full flexibility. Leather socks like with a side zip, that's the flexible. So blind docking, trying shoes without looking at your feet, you are wearing them and talking to someone else, with your partner or with your family members or anyone else. So for blind docking, use CB-Dock or set a large grid box in AutoDock Vina, that you have to give the dimensions to the AutoDock Vina, and it will blindly dock it and give you the binding poses and also the maximum minimum energy of binding pose. So protein preparation for docking, Proper protein preparation is crucial for accurate docking. Steps: we have to remove water molecules, fix missing atoms and residues, and add polar hydrogens. That we will see in our workflow when we go to the softwares. I'll show you that how you can add charges, add polar hydrogens, or you have to add non-polar, then you can add non-polar hydrogen, but generally we used to add polar hydrogens. And how you can add Coleman charges or gestures, and also we how can we define the grid. We have to give the grid dimensions. We set a grid box and the software generates the dimension, that xyz dimensions, xyz sizes, and it will give the whole dimension. That we make a separate file that you can say it like the configuration file to the AutoDock Vina. You give it to AutoDock Vina, or in the PyRx you set the uh, grid box and it generates the grid dimensions in the software, and all the things, ligand preparation, protein preparation, and grid dimensions, and everything is done in the single software, that is PyRx. Also you can do everything in the molecular operating environment like MOE, but in AutoDock Vina that's the accurate one, as per my thoughts, and as per many of the researchers thought that AutoDock Vina is the accurate one than anyone else. So you have to give the grid dimensions. So for the protein preparation, the steps like remove water molecule, fix missing atoms or residues, add polar hydrogens, add Coleman charges, define active site grids, save as PDBQT. The format for the docking is mandatory, or the format of your file is mandatory, that you have saved the file in which format. You have saved it in the PDB, but the Vina you are going to run, it reads only PDBQT files. You save it in the SDF format, but Vina will not read it. Vina will not even pick it from your suggested or given path folder of your laptop. Vina don't even pick it because it's not the format that Vina reads. So the main is format for the docking. The prepared protein structure and also the ligands are saved in the PDBQT for AutoDock, compatible with the choosing docking software, and also compatible if you are using the molecular operating environment, then you have to save it in the .mol or MOE format. So protein preparation is like cleaning a workspace before starting a project. You remove unnecessary items, water molecules, and align the tool charges, and then mark where you will work active site. So using MGL tools, basically I will guide you on the MGL tools, that how you can prepare a protein and how you can prepare a ligand in the MGL tools that offer the few steps like loading protein, editing, deleting water molecule, adding hydrogens, adding charges, and saving it as a PDBQT, and also setting the grid box for getting the grid dimensions. So the second one step is ligand preparation for docking. Ligand must also be cleaned and optimized. We have to download it from PubChem or Zinc or Drug Bank, and a lot of the servers that we have discussed before. We have to download ligand from them and then convert it using OpenBabel, or we can also use AutoDock tool from the MGL tools to optimize, minimize, or convert it to the PDBQT format. for adding hydrogen charges or energy minimization. We can also done it from the software like Avogadro's. We will use it, and also adding hydrogen and charges using AutoDock tools, as we I have discussed before, the MGL tools. So, and the at the end saving it as a ligand.pdbqt. So preparing a ligand is like sharpening a pencil before writing. A dull pencil, non-optimized ligand, gives a poor result. So that's the ligand preparation. Ligand preparation is a little bit short, but it's tricky one than the protein. Popular molecular docking tools: AutoDock Vina, as I have discussed before, it's the accurate one and also the free one and powerful one. And free one and powerful one, AutoDock Vina. AutoDock 4 is also used, and that relies on the genetic algorithm, and PyRx, GUI based for the beginners friendly. If you are a beginner, you first go to the PyRx and then go to the AutoDock Vina, or then to the Gold or Schrodinger. Schrodinger is too complex, and you also need uh, big machinery like um, big man for the big work. So you have to purchase a Linux software, a Linux server, and the big servers to run Schrodinger. But AutoDock, Pyrex, Gold, AutoDock 4, and also many of the web servers like HeadDock, and a bit more as we go further in our um, lectures like nanoinformatics, and for the nanoparticles docking. There you will learn how we can prepare a nanoparticle in our laptop, on our desktop, and how we can dock them in a protein or in our suggested biomolecule or with any other molecule we are going to dock them. So these are the tools. We have to just import protein, import ligand, define search space, and then running Vina and exporting results. These are the basic popular molecular doc. So web servers, as I have discussed before, that there are also the web servers. There not only the software but also the web servers. There you will only give the protein, give the your ligands, and also some of the servers suggest you to provide the um, grid dimensions, that where you want to dock them. But most of the servers like SwissDock, they dock on their own. They find out the binding sites and dock on them and then give you the results. So the web servers for docking, web-based tools are excellent for user without strong hardware. If you have a laptop that not support even hardware, but my first laptop was Core i3 with the fourth generation, but I built it on my own with putting many things in its machinery, like I put their hardware name was, and there is hardware name. I put the hardware, extra hardware, and also the hardware that 500 GB. I extracted the CD-ROM and put the caddy and caddy here in the hardware, and also the fast um, that the hardware name don't coming to my mind, but you better know, everyone. So SwissDock, DockThor CB-Dock, Permit, and Quick Vina W, and these are the web servers. There we have to upload protein, upload ligand, and select accurate mode. We have to do these things on the, then we have to submit, then we have to just download results, and the results will be in two formats, like in the, as we get the results from any, from the software or any web servers, we get two kinds of the files. The one file is the PDBQT or PDB file or mol file that can be visualized at how the bonding made between the protein and the ligand. And the second one file is maybe the Excel file or the Word file that or the text file that giving you the binding energies. That the first binding pose has a binding energy minus 8.8. That's a good one. And the third one is the visualization tools. Fourth one step is the visualization tools. First we have to prepare protein, then prepare ligand, then we have to give them to the docking tools or web servers, and then the number fourth one is the visualization. The visualization help interpret results, that how the bonding is made. And there are four major tools that can be used for visualizing. Number one is PyMol. That's a good one and free one. And number second is Discovery Studio. That's also a good one. PyRx. I have used the PyRx, but I like most the PyMol and Discovery Studio. These are user friendly. And the VMD Visual Molecular Dynamics, a program for displaying, animating, and analyzing large biomolecular systems. So there you can analyze hydrogen bonds, hydrophobic interactions, binding pocket fit, compare binding poses, and compare Vina source. It's like examining how well a shoe fits your foot, tightness, shape, angle. And comprehensive molecular docking workflow, protein retrieval, protein preparation, ligand retrieval, ligand preparation, docking, visualization, admit filtering. Admit filtering is the major thing that you can use the web servers like Admit Lab 2.0 or SwissDock for the prediction of the admit properties of the ligand, like absorption, distribution, and metabolism, excretion, and toxicity of that. That would be optional. Okay. Docking is like choosing a perfect mobile phone cover. You check if the cutouts match, the fit is correct, and the grip feels right. Molecular docking uses the same idea but at a microscopic level. So that was the lecture for the molecular docking basics, and I'll upload next video on the guidance of how you can use different tools for preparation of protein and ligand, and we can step in the lab of computer, the visual world, as it's roaming around the visual worlds. So don't forget to like, comment, and subscribe, and follow us on Facebook. It's Hammad Bashir Khan, in a bracket BiochemVerse, and also on the YouTube it's BiochemVerse by Hammad. And also follow us on our WhatsApp channel. There you will get the updates about every lecture on the Facebook or YouTube. It's uploaded. So thank you so much, and again sorry for missing lectures and so much vacations. Thank you so much. Allah Hafiz.